We're going to take a look at how muscles contract in this lecture. The sliding filament model is what we use to describe how muscles contract and shorten. So that's really important to know. So the sliding filament model. So when muscles contract, they do shorten. And it's important to note that the thick and the thin filaments, so the myosin and the actin, they actually do not shorten at all. So what's happening is the thin filaments are going to slide over the top of the thick filaments, and that's what's going to cause the muscle to shorten and contract. And how all of this happens, how the thin filaments slide over the top of the thick filaments, is due to this cross-bridge cycle. So we're going to go over that in a minute, and that's really important to know as well. So you need to know the sliding filament model, which is just saying that the thin filaments slide over the top of the thick filaments, and that's what causes the muscle to contract and shorten. And that is due to this cross-bridge cycle. That's what makes the thin filaments slide over the thick filaments. All right, and then the other thing that I want you to know that would be easy to ask on the next exam is what's going to shorten during contraction when we look at our sarcomere. So the way that I remember this is I remember that the word his, so if I take the first letters here, so the word his is a short word, and so all of these are going to shorten. So the H zone, the I band, and the whole sarcomere itself, those are all going to shorten. But the A band does not shorten. Okay, so the A band is the length of the myosin. And remember that we said the myosin, or the thick filament, does not shorten. So it's going to stay the same length, right? Okay, so let's just take a look at some pictures of this. So we've got a sarcomere up here on the top. We've got a Z line. Here's the other Z line. So in between here, we've got our sarcomere. And this is the muscle at rest. So it hasn't started contracting yet. And when it does contract down here on the bottom, you can see that the whole sarcomere has shortened. So from Z line to Z line, that has shortened. And then your H zone. Okay, remember here in the middle where there's just the thick filament and no overlap with the thin filament there, that's shortened. And then the I bands, the light bands, those have also shortened. Okay, but that A band, which is the length of the thick filament or the myosin, that stays the same length. So that is not shortened at all. And then here's an actual microscope view of the sarcomere. And we can see the sarcomere is relaxed here on the top. Muscle has not started contracting yet. So from Z line to Z line, that's going to be one sarcomere. And then on the bottom, our muscle has contracted. So you can see the sarcomere has shortened. And then the H zone here in the middle, that's shortened. And then the I bands on either side here, those light bands, they have shortened. But if you look at the A band, this dark band here, that's the length of the myosin, or the thick filament. That has not changed. It is not shortened. It's stayed the same. And then this little link here, if you want to watch that at home, it's just a little animation, but it shows the sarcomere relaxed and then contracting, and so it actually shows all of these things shortening as the muscle is contracting. So it's pretty good to watch. The crossbridge cycle is how we actually get the thin filaments to slide over the top of the thick filaments and get that sliding filament model to work and get the muscle to contract and shorten. So this crossbridge cycle is really important to know. And I've got everything written down that happens in the crossbridge cycle, and we're actually going to look at some pictures here to explain this. But just a couple of things that are important. We do need some calcium for the crossbridge cycle to happen. And if you remember where calcium is stored in the muscle cell. So remember that's stored in the sarcoplasmic reticulum, which remember that's actually the smooth ER. And then we're also going to need some ATP or energy for the crossbridge cycle to happen. So definitely need energy for the muscle to contract. And we're going to be talking about this power stroke, and we'll also be talking about rigor as well. So, and rigor just means rigid. Okay, so we'll talk about that as well. All right, so let's start with our crossbridge cycle up here. And we're going to have our thin filament or actin. And then right below that, we've got the myosin or the thick filament. And to start this crossbridge cycle, we're going to be releasing calcium from the sarcoplasmic reticulum. And the calcium is kind of hard to see, but it's in green. And the calcium is going to bind to the protein called troponin that's on the actin or the thin filament. And when calcium binds to troponin, that's going to move the tropomyosin rope that's on the thin filament out of the way, and that'll expose these little black binding spots on the thin filament for the myosin heads. Okay, because when the tropomyosin is over those binding sites, the myosin cross bridges or heads there cannot bind to the thin filaments. But once we move the tropomyosin out of the way, then the myosin heads can go ahead and grab onto the thin filaments. Okay, so once we've got the calcium bound, and now we've got those binding spots on the thin filaments exposed for the cross bridges or the myosin heads, those myosin heads are already ready to go. And so the way I think about those myosin heads is they're kind of like little slingshots. Okay, so they've already taken the ATP, and you can see that they've chopped off one of the phosphate groups. Okay, and they're still bound there. They haven't let go of that yet. And it's kind of like you've taken the slingshot and you've pulled it back. Okay, so they've cocked the myosin head back, and it's just sitting there waiting. Okay, and as soon as those binding sites on the thin filaments become available, then that myosin head is going to reach up and grab onto the thin filament and grab it. Okay, and once it does that, then it's going to rotate, and it's going to do what's called a power stroke. Okay, so it's just going to reach up, grab onto the thin filament or the actin, and then it's just going to rotate and do a power stroke, and it's going to move that thin filament towards the M line, towards the middle of the sarcomere. Okay, and so if you look at my fingers here, my thumbs would be like the Z lines, and that would be one sarcomere, and then my fingers would be like the thin filaments here. And what's going to happen is the thin filaments are just going to slide towards the M line, and you can see how the Z lines are going to move closer together, and then the sarcomere is going to shorten. Okay, so we're just going to move the thin filaments towards that M line, and that'll shorten the sarcomere. Okay, so our little cross bridge or myosin head, like a slingshot, It's just going to grab onto the thin filament. It's just going to rotate and pull that thin filament towards the middle of the sarcomere. Okay, and it will just stay in that position, and that's called rigor. Okay, so it just stays there like this until ATP binds to the myosin heads, and then it can let go of the thin filament. Okay, but it will just stay there, bound and holding on to that thin filament until the ATP binds. So we call that rigor because it'll hold the thin filaments in that contracted position until the ATP binds, and then it can let go. Okay, and then once the ATP binds, the enzyme that's on the myosin head, that ATPase, will chop off the phosphate group, release the energy, and then we're just going to take that myosin head again and we're going to pull the slingshot back. So we cock the myosin head back again, and then if the tropomyosin rope is still out of the way, the binding sites are still exposed on the thin filament, then we'll just reach up, grab onto the thin filament again, do another power stroke, and then just do another cross-bridge cycle. And so we'll just go around and do this whole thing again. Okay, so that's the cross-bridge cycle and what's happening there. And there's a really good little animation tutorial. I'm sorry, my little video here is in the way, but on the PowerPoint you can click on that, and it's just real short, but it goes through and walks you through this cross-bridge cycle. So I would definitely watch that at home because it explains this, and there will definitely be questions on the cross-bridge cycle on the exam. So that's really important to know. The other thing to explain, rigor mortis. So probably all have heard of this. So when somebody dies or an animal dies, after a while, the body gets really stiff and rigid. All the muscles contract. So what happens is as the muscle cells start to break down, the sarcoplasmic reticulum starts to break down and actually releases calcium into the sarcoplasm or the cytoplasm, and that will actually bind to the troponin, lift the tropomyosin rope out of the way, and because the myosin heads, you've already pulled those slingshots back and they're just ready to go, and so they're like, oh, you know, binding sites are available, so we're just going to reach up, grab onto those thin filaments, we're going to do our power stroke and slide those thin filaments towards the middle of the sarcomere and shorten the sarcomere, and so the muscles will contract. But the problem is that because you're dead, you can't produce any more ATP, and your cells don't have stores of ATP in them. And so what happens is you're going to be stuck in this rigor step because you don't have ATP to bind to the myosin heads to make them let go of the thin filament. And so you can't relax the muscle at all. They just stay contracted. Okay, so that's rigor mortis. But after a while, the rigor mortis will go away, and then the muscles do relax, and then you can bend, you know, the person's arms and legs again, because while they're in the rigor mortis, you cannot bend them because they are just so rigid and stiff. You'd have to pretty much break bones to get them to bend. But once things, you know, keep degrading inside the cells, inside the muscle cells, the actin and myosin start to disintegrate, and then that's what causes the muscles to actually relax, and then rigor mortis will go away. Okay, so as things start to disintegrate more. So that's just what happens in rigor mortis, so if you're interested. Okay. So the importance of the cross-bridge cycle is that the sarcomere is going to shorten. And remember that in our myofibril, we've got these sarcomeres just lined up end to end along the whole length of the myofibril, which runs the whole length of the muscle cell. And so all of these sarcomeres will be shortening, and then the whole myofibril will shorten, and all of these myofibrils in the muscle cell will be shortening. And then remember that the muscle, the whole muscle itself, all of those muscle fibers will be shortening, and when the muscle shortens itself, it will generate force, and then that will pull on the tendon, which is attached to the bone, and that will allow you to move the limb, and then you can, lift something, or, whatever you're going to be doing with that. So that's why these cross-bridge cycle is really important, to be able to slide the thin filaments over the thick filaments, shorten the sarcomeres, and that'll cause the muscle to contract, and then you can generate force, pull on the tendon, and move your bone, and move.